Hello students, myself Dr. Sachin Kapur, and today we will be discussing the topic of cleavage or cellulation. In our previous lecture we discussed about fertilization. Now let's understand what is cleavage. As we can see in this diagram, we know that fertilization occurs in the ampulla region of the fallopian tube, more precisely at the ampullary isthmic junction. As a result of fertilization, a single-celled diploid structure called zygote is formed. So, here I have shown zygote. With the red color, we have shown here zona pellucida. So, zona pellucida layer surrounds the fertilized egg or the zygote. This zygote will undergo cleavage divisions to form a solid mulberry-like structure called morula. That is what is cleavage, those mitotic divisions. Cleavage is nearly similar to mitosis, but there are certain differences which we will discuss. So, what is cleavage? It is repeated rhythmic mitotic division which converts single-celled zygote into a multicellular structure called morula. So, zygote is a single cell and morula is multicellular structure. It has 16 cells. It is without any central cavity. When the cavity starts developing, the cavity called blastocele. So, when the cavity develops, then this structure is called blastula or blastocyst. The term blastocyst we use in case of mammals. So, let us see what is cleavage. As this diagram explains that when cleavage happens in the zygote, when the first cleavage happens, two daughter cells which are called blastomeres are formed. In humans, the first cleavage division occurs 30 hours after fertilization. So, we will write here, in humans, the first cleavage occurs 30 hours after fertilization and two daughter cells called blastomeres are formed. The second cleavage division occurs 10 hours after the first and 4 daughter cells are formed. So, we will write second cleavage division occurs 10 hours after first and 4 daughter cells or the 4 blastomeres are formed. The next cleavage division or the third cleavage division is completed by the end of third day with respect to fertilization, and as a result of that you get a structure which has 8 cells. So, first cleavage 30 hours after fertilization, 2 blastomeres formed. Second cleavage division 10 hours after the first and 4 daughter cells called the blastomeres are formed. Third cleavage is completed by the end of third day with respect to fertilization, and this structure with 8 blastomeres is formed. The next cleavage division results in the formation of a solid mulberry-like structure which is called morula. So, I will write for morula, what is morula? It is solid mulberry like structure without any central cavity. Morula has 16 cells. So, the 16th cell stage is what is called morula. This dividing zygote, that is the zygote which is undergoing cleavage, it keeps moving towards the uterus. We know that the entire fallopian tube is ciliated. So, the dividing zygote keeps moving down towards uterus, because ultimately the implantation, the attachment of this embryo is going to happen in the uterus. Zygote is a single-celled embryo, right? When you are asked that what's an embryo, even zygote is an embryo, a single-celled embryo, right? So, the dividing zygote keeps moving towards the uterus. The size of morula and zygote is nearly similar. Why? Because this layer zona pellucida a glycoproteinaceous layer it is still intact. Till the time zona pellucida is intact, the size of that structure cannot increase because zona pellucida is not allowing that. Now, why the zona pellucida does not dissolve in the fallopian tube itself? The reason is the presence of zona pellucida ensures that implantation, that is attachment of the blastocyst, should happen at a proper site, that is uterus. Uterus is designed to carry on the pregnancy. If zona pellucida dissolves in the fallopian tube itself, then this structure will attach with the wall of fallopian tube, resulting in a very dangerous situation that is called tubal pregnancy. It's a case of ectopic pregnancy, means a pregnancy which is not in the uterus, that is called ectopic. So, what is the significance of zona pellucida here? Please remember that zona pellucida will ensure that implantation should happen at proper site. And what is the proper site? Uterus. If zona pellucida dissolves in fallopian tube, then the structure will attach with the wall of fallopian tube, resulting in tubal pregnancy. The fallopian tubes can burst also, resulting in excessive bleeding, and she can die also, right? So, as a result of cleavage, we get a solid mulberry-like structure that is called morula. It takes 4 days for the morula to reach uterus. The dividing zygote takes 4 days to reach the uterus. And when it reaches uterus, the uterus is in the secretory phase of the menstrual cycle. We know that if the cycle is of 28 days, ovulation happens on day 14, and after that, if the spermatozoa are there in the female tract, fertilization will happen and zygote is formed. So, the zygote undergoes cleavage divisions and moves towards the uterus. After ovulation, the uterus is in the secretory phase of the cycle. That means uterine lumen is full of nutritive substances. The endometrial glands are secreting the nutritive substances and they are maintaining the uterus in a secretory stage, a favorable stage. Why? Because now the next step is going to be implantation. So, the morula which has arrived in the uterus should get a favorable environment. Uterus should not contract. If the uterus contracts in this situation, the structure will be expelled out. So, progesterone hormone makes the uterus in the secretory phase and it ensures that uterine muscles, smooth muscles should not contract, and the endometrial glands should secrete nutritive substances so that a complete nutritive bed is prepared in the uterus. The endometrium is thickened so that implantation can happen. Now, the morula is going to float in the uterine lumen for another 3 days. It absorbs nutritive substances, it develops a central cavity called blastocele, and the cells of morula rearrange themselves into an outer layer called trophoblast, and few cells are attached at one corner, that's called embryoblast or ICM. The ICM stands for inner cell mass. The trophoblast has no role in the formation of embryo proper. The trophoblast is involved in the formation of extra embryonic membranes and placenta. It's going to provide nutrition to the embryo. The embryo proper develops from embryoblast or the cells of the inner cell mass. The cavity is blastocele, the structure is blastula. So, morula keeps on floating in the uterus for nearly 3 days, absorbs nutritive substances and changes into blastula. That was about cleavage. Now, let us see what are the differences between cleavage and a typical mitosis. Number 1, cleavage divisions are very quick and the growth phase is very short. The size of the daughter cells doesn't increase. Number 2, nuclear cytoplasmic ratio increases during cleavage divisions. So, cleavage divisions are quick as compared to mitosis, growth phase is very short, the size of the daughter cells doesn't increase, right? And number 3, that the nuclear cytoplasmic ratio increases during cleavage. So, that was about cleavage. In brief, what is cleavage? The zygote will be converted into a multicellular structure called morula by the process of cleavage. The first cleavage is going to start 30 hours after fertilization, then the next will be 10 hours after the completion of the first, then the third cleavage is completed by the end of third day with respect to fertilization. The dividing zygote is moving towards the uterus because ultimately it has to implant in the uterus. And the next process which is going to happen after this is the implantation or the nidation. Implantation is the attachment of this developing blastocyst with the wall of the uterus. In our forthcoming lectures, we will discuss about implantation in detail. Thank you.